Welcome, esteemed researchers, to an exploration of one of biology's most profound and promising frontiers, stem cell science. This guide delves into the fundamental properties, research methodologies, and transformative potential of stem cells, offering insights crucial for advancing our understanding of health and disease. Prepare to uncover the intricate mechanisms that govern life's most basic building blocks. Stem cells are extraordinary biological entities distinguished by two defining characteristics: their capacity for self-renewal and their ability to differentiate into specialized cell types. In vitro, these cells can proliferate extensively, maintaining an undifferentiated state. This unique combination of properties makes them indispensable for tissue maintenance, repair, and regeneration throughout an organism's lifespan. Historically, the existence of stem cells was inferred long before their direct identification. Early evidence emerged from observations in hematology, particularly bone marrow transplantation. Researchers noted that a small volume of transplanted bone marrow could fully reconstitute the entire hematopoietic system of a recipient, including red blood cells, white blood cells, and platelets. This remarkable regenerative capacity strongly suggested the presence of a foundational cell population capable of generating all blood cell lineages. This foundational understanding led to the categorization of stem cells based on their developmental plasticity, or potency. This classification system helps researchers define the range of cell types a stem cell can generate, guiding experimental design and therapeutic applications. Understanding these distinctions is paramount for precise research and clinical translation. Totipotent stem cells represent the highest degree of developmental plasticity. A single totipotent cell possesses the unique ability to differentiate into all cell types of an organism, including both embryonic tissues and extra-embryonic tissues like the placenta and umbilical cord. This complete developmental capacity is exemplified by the zygote, the single cell formed immediately after fertilization. Following fertilization, the initial few cell divisions of the zygote produce more totipotent cells. These early blastomeres retain the full potential to form an entire organism. However, this totipotent state is transient, lasting only for the first few days of embryonic development. Researchers study these early stages to understand fundamental processes of cell fate determination and developmental programming. As embryonic development progresses, totipotent cells give rise to pluripotent stem cells. These cells can differentiate into any cell type derived from the three germ layers: ectoderm, mesoderm, and endoderm, which form all the tissues and organs of the body. However, unlike totipotent cells, pluripotent cells cannot form extra-embryonic tissues necessary for fetal development. Embryonic stem cells, ESCs, derived from the inner cell mass of a blastocyst, are the quintessential example of pluripotent stem cells. Their remarkable capacity for indefinite self-renewal and broad differentiation potential makes them invaluable research tools. They serve as models for studying early human development, disease mechanisms, and drug screening. Multipotent stem cells possess a more restricted differentiation potential compared to pluripotent cells. They can differentiate into multiple cell types, but typically only within a specific lineage or tissue. For instance, hematopoietic stem cells, HSCs, are multipotent, capable of generating all types of blood cells, but not nerve cells or muscle cells. These cells play a crucial role in tissue homeostasis, repair, and regeneration throughout life. Researchers are intensely focused on identifying, isolating, and characterizing multipotent stem cells from various adult tissues. Understanding their niche, signaling pathways, and regenerative capacity is key to developing targeted therapies for tissue damage and degenerative diseases. Adult stem cells, also known as somatic stem cells, are multipotent stem cells found in various differentiated tissues throughout the body. They reside in specialized microenvironments, or niches, where they remain quiescent until activated by injury or the need for tissue turnover. Examples include mesenchymal stem cells, MSCs, neural stem cells, NSCs, and muscle satellite cells. Recent groundbreaking research has revealed that adult somatic cells can be reprogrammed into an embryonic stem cell-like state, generating induced pluripotent stem cells, iPSCs. This revolutionary discovery, pioneered by Shinya Yamanaka, bypasses the ethical concerns associated with ESCs while offering a patient-specific platform for disease modeling and regenerative medicine. iPSCs have transformed the landscape of stem cell research, providing an unprecedented tool for studying human biology in vitro. The study of stem cells is not merely an academic pursuit. It is a research imperative with profound implications for human health. Understanding stem cell biology provides fundamental insights into developmental processes, offering clues to the origins of birth defects and congenital disorders. Moreover, aberrations in stem cell function are implicated in numerous pathologies, including cancer and degenerative diseases. Stem cells serve as powerful in vitro models for investigating complex biological phenomena. Researchers utilize them to dissect the molecular mechanisms underlying cell differentiation, tissue formation, and disease progression. This allows for the study of human-specific cellular processes that are difficult or impossible to observe in vivo, providing a controlled environment for experimental manipulation. One of the most significant applications of stem cell research is the creation of patient-specific disease models. By generating iPSCs from individuals with genetic disorders, researchers can differentiate these cells into affected cell types, such as neurons for neurological diseases or cardiomyocytes for cardiac conditions. This enables the study of disease pathogenesis in a relevant human cellular context, facilitating the identification of novel therapeutic targets. These models are invaluable for high-throughput drug screening, allowing researchers to test thousands of compounds for efficacy and toxicity. This accelerates the drug discovery pipeline, potentially leading to more effective and safer treatments. Furthermore, patient-derived stem cell models can be used to understand individual variability in drug response, paving the way for personalized medicine. The most exciting and transformative potential of stem cells lies in regenerative medicine, specifically cell therapy. This approach aims to treat diseases by replacing damaged or dysfunctional tissues with healthy, functional cells derived from stem cells. While organ transplantation currently addresses tissue failure, the severe shortage of donor organs necessitates alternative strategies. Researchers are actively exploring stem cell-based therapies for a wide array of debilitating conditions. Neurological disorders such as Parkinson's disease, Alzheimer's disease, stroke, and spinal cord injury are prime targets, where stem cells could potentially replace lost neurons or support neural repair. Similarly, chronic conditions like heart disease, diabetes, and osteoarthritis could benefit from the regenerative capacity of stem cells, offering hope for restoring tissue function and improving patient quality of life. Aging is a complex biological process characterized by a progressive decline in physiological function and an increased susceptibility to disease. A major contributing factor to this decline is the impaired function of our endogenous stem cell populations. As organisms age, their stem cells exhibit a reduced capacity for self-renewal and differentiation, compromising the body's ability to repair and regenerate tissues. Researchers are actively investigating the mechanisms underlying age-related stem cell dysfunction. Several hypotheses are being explored. Stem cell burnout suggests that repeated divisions over a lifetime deplete the stem cell pool. Accumulation of DNA damage, oxidative stress, and epigenetic alterations within stem cells can impair their function. Furthermore, changes in the stem cell niche, the microenvironment that supports stem cell maintenance and activity, can send altered signals, leading to reduced stem cell activity.